Good day everyone. Welcome to another lecture video in molecular biology. Today, we will be talking about cellular respiration. So how electron transport is used to trap chemical bond energy. The molecule at left is oxidized by the loss of an electron. The molecule at right is reduced by gaining an electron. Having seen in the previous lecture video that ATP is the one common energy denominator by which most cellular machines are powered, we must ask how this energy is captured from fuel substrates. This question directs us to an important generalization. All cells obtain their chemical energy requirements from oxidation-reduction reactions. This means that in the degradation of fuel molecules, hydrogen atoms, electrons and protons are passed from electron donors to electron acceptors with a release of energy. A portion of this energy can be trapped and used to form the high energy bonds of molecules such as ATP. An oxidation reduction or redox reaction involves a transfer of electrons from an electron donor, the reducing agent, to an electron acceptor, the oxidizing agent. As soon as the electron donor loses its electrons, it becomes oxidized. As soon as the electron acceptor accepts electrons, it becomes reduced. In other words, a reducing agent becomes oxidized when it reduces another compound, and an oxidizing agent becomes reduced when it oxidizes another compound. Thus, for every oxidation, there must be a corresponding reduction. Aerobic metabolism is more familiarly called cellular respiration, defined as the oxidation of fuel molecules to produce energy with molecular oxygen as the final electron acceptor. We emphasize that oxidation of fuel molecules describes the removal of electrons and not the direct combination of molecular oxygen with fuel molecules. Let us look at this process in general before considering it in more detail. Hans Krebs is a British biochemist who contributed so much to our understanding of respiration. He described three stages in the complete oxidation of fuel molecules to carbon dioxide and water. In stage 1, food passing through the intestinal tract are digested into small molecules that can be absorbed into the circulation. There is no useful energy yield during digestion. In stage 2, also called glycolysis, most of the digested food is converted into 2,3 carbon units of pyruvic acid in the cell cytoplasm. The pyruvic acid molecules then enter mitochondria, wherein in another reaction they join with a coenzyme A or CoA to form acetyl-coenzyme A or Acetyl-Coa. Some ATP is generated in stage 2, but the yield is small compared with that obtained in stage 3 of the respiration. In stage 3, the final oxidation of fuel molecules occurs, with a large yield of ATP. This stage takes place entirely in mitochondria. Acetyl-CoA is channeled into the Krebs cycle, where the acetyl group is completely oxidized to carbon dioxide. Electrons released from acetyl groups are transferred to special carriers that pass them to electron acceptor compounds in the electron transport chain. At the end of the chain, the electrons and the protons accompanying them are accepted by molecular oxygen to form water. We begin our journey through the stages of respiration with glycolysis. a nearly universal pathway in living organisms that converts glucose into pyruvic acid. In a series of reactions occurring in the cell cytosol, glucose and other 6-carbon monosaccharides are split into 3-carbon molecule called pyruvic acid. A single oxidation occurs during glycolysis, and each molecule of glucose yields 2 molecules of ATP. In this pathway, the carbohydrate molecule is phosphorylated twice by ATP. First to glucose phosphate, which is not shown in the figure, and then to fructose biphosphate. The fuel has now been primed with phosphate groups in this uphill portion of glycolysis and is sufficiently reactive to enable subsequent reactions to proceed. This is a kind of deficit financing required for an ultimate energy return many times greater than the original energy investment. In the downhill portion of glycolysis, fructose biphosphate is cleaved into 2 3-carbon sugars, which undergo an oxidation, or when electrons are removed, with the electrons and one of the hydrogen ions being accepted by nicotinamide adenine dinucleotide, or NAD+, which is a derivative of the vitamin niacin, to produce a reduced form called NADH. So NADH serves as a carrier molecule to convey high-energy electrons to the final electron transport chain, where ATP is produced. The 2 3-carbon sugars next undergo a series of reactions, ending with the formation of 2 molecules of pyruvic acid, as in this figure. In 2 of these steps, a molecule of ATP is produced. In other words, each 3-carbon sugar yields 2 ATP molecules, and since there are 2 3-carbon sugars, 4 ATP molecules are generated. Recalling that 2 ATP molecules were used to prime the glucose initially, the net yield at this point is 2 ATP molecules. The 10 enzymatically catalyzed reactions in glycolysis can be summarized as glucose plus 2 molecules of ADP plus 2 molecules of inorganic phosphates plus 2 molecules of NAD+, which will form 2 pyruvic acid plus 2 NADH plus 2 ATP. In aerobic metabolism, the 2 molecules of pyruvic acid formed during glycolysis enter a mitochondrion. There, each molecule is oxidized, and one of the carbons is released as carbon dioxide. The 2-carbon residue condenses with coenzyme A or CoA to form acetyl coenzyme A or Acetyl-Coa and an NADH molecule, which is also produced in this reaction. Acetyl-Coa is a critically important compound. Its oxidation in the Krebs cycle provides energized electrons to generate ATP, and it is also a crucial intermediate in lipid metabolism. Krebs cycle and oxidation of acetyl-CoA. Krebs cycle degradation or oxidation of the 2-carbon acetyl groups of acetyl-CoA occurs within the mitochondrial matrix in a cyclic sequence. It is also called citric acid cycle and tricarboxylic acid cycle, or TCA cycle. Acetyl coenzyme condenses with a 4-carbon acid, oxaloacetic acid, releasing coenzyme A to react again with more pyruvic acid. Through a cyclic series of reactions in the Krebs cycle, the 2 carbons from the acetyl group are released as carbon dioxide. and oxaloacetic acid is regenerated. Hydrogen ions and electrons in the oxidations transfer to NAD and to FAD, or what we call as flavine adenine dinucleotide, which is another electron acceptor, and a pyrophosphate bond is generated in the form of guanosine triphosphate, or GTP. This high-energy phosphate readily transfers to ADP to form ATP. Thus, the overall products of the Krebs cycle are carbon dioxide, ATP, NADH, and FADH2. As in this reaction, the molecules of NADH and FADH2 formed yield 11 molecules of ATP when oxidized in the electron transport chain. The other molecules in the cycle behave as intermediate reactants and products which are continuously regenerated as the cycle turns. That's the end of the lecture video. I hope I will see you soon. Thank you for watching. Have a great day and stay safe, everyone. Goodbye.